Hey folks, this is an introduction to the microscope, and at Finger Lakes Community College we have two different kinds of microscopes, probably the same brand name, but there's one that's a little bit more modern looking, but they are very, very similar. So if you learn how to use one, just the buttons are a little bit different on the other. So let's go through a couple things of this microscope. First of all, when you carry a microscope, you carry it with the handle and you grab it underneath. Okay, don't ever grab it with one hand or over lean it over. Some of these things may be loose and they'll fall on the floor and you owe me $10, so don't do that. Okay, so when you carry the microscope, by the arm and underneath, always two hands when you carry the microscope. Okay, now this is the eyepiece over here, and the eyepiece for this one actually has two different ones. So for now we're going to turn the one so it's level with the other. The first thing you do. And it says a 10x right there on there. We'll talk about that in a bit. Okay? The first thing you should see when you do the microscope is, was it stored correctly? This one was, because it's on the 4x power right there. On these objective lenses, this is at 4x. That's how it should have been stored. This one was stored incorrectly. Somebody put it on there. So gently turn the wheel, don't grab here, turn the wheel, put it back to the red, the 4x. Okay? That's how it should be stored, so when you're done with the microscope, you do it correctly and store it back here. Alright, well let me show you a few things. One is, there's an on switch somewhere that turns on the light. So see, this light is turning on when I turn this on switch on. The other microscope has an on switch that's somewhere else, like in the front, and then the wheel to make it bright and dim is on this side. So they might be in different spots. This one has the on switch right on the side, and it has the dimmer switch for the light right there. As long as we're talking about the light, see if you can get underneath here, and you'll see there's a little diaphragm like in a camera, like an aperture. And this aperture means you could change the brightness of the light on the specimen just by turning this dial all the way open on the left, all the way closed on the right. My particular preference, put it in the middle. Then you could change the brightness. I usually dial this up to like 3 on this one and about 8 on this one, all right? But when you dial up the brightness, then you could not touch that anymore, and then just do like a fine tuning of the brightness right there at the scope. Okay? All right. We are obviously going to look in here at a specimen that's right here on this edge here. Let me show you a couple more things that are important. See this piece right over here? This, when you have expensive scopes like this, you don't have to move the slide on your own. You actually turn this wheel to move this way and turn the other wheel to move it back and forth like that. Isn't that cool? So when you put a microscope slide there, it just fits right in that little groove, and then we'll do that. Let me just grab a microscope slide really, really quick. Like this one is the letter E. Watch how I do this. I'm going to stick the letter E in there. Watch. I open this up. I put the letter E in there, and then I hold that back. Now it's being held in place. See where the little bright light is? I could visually center that thing right there in the middle. Isn't that nice? So the letter E is right in the middle of the brightness. Okay? And if we look through the scope right now, we should be able to see the letter E. Now, one more most important thing, I can't stress this enough, is this spot right over here. This is the focus knobs. The outer ring is called course adjustment, and you never ever use this unless you have the 4x one on there. Promise. Never touch the course unless low power is on. The first one we call low power. The next one we'll probably have is called like medium power. The first one's like low power or scanning. The next one is low power, and then the last one is high power. For our class, we're not going to use the last one. You need actually a drop of oil as an immersion to get that lens to even work. Okay? So if you take another upper level bio course, you're going to use the oil immersion one. So for now, we're only going to use the red for low power or scanning, and then like a medium power one, and the blue for the high power. Okay, so we go back to low power because we're not going to touch the focus knob. Now look what happens when I turn this knob. It goes from way up here, that's as high as it gets, and moves down. So we're actually changing where the object is next to the lens, and that's how we focus. So this is our focus knob, and it's called coarse adjustment. You never ever use this unless you're on the 4 power, the low power. The middle knob is called fine adjustments, and that's the one that you're going to use from then on. So once you get close to being in focus with the big knob, you're never going to touch the big knob again. Then you touch the inner knob that moves it ever so slightly, and then you get it in fine sharpness. Now, this scope comes with 2. So there's a couple different techniques you can use for this, but depending on how wide your eyeballs are, you know, you're either going to be way over here or you're going to be close over here if your eyes are close together. So you find one where you comfortably can look in both. Now, different teachers have different techniques. Your teacher might have a different technique. My technique is look with your right eye in this one and then focus it with this knob. So I'm looking in with just one and I'm focusing it with just this one. And then I am seeing the little letter all in focus right there. Okay? And when I get the thing in focus, okay, then once I know this is in focus, then I turn this knob so my left eye is also in focus on the same object. And when I get them both in focus, I never touch it again. Then I know my left eye and my right eye are both in focus, and then I can just touch the fine adjustment from that on. Okay, so that's the part of the scope. If you read the lab, you'll see that there's a list of all these different pieces of the scope, but that's how you do it. The most important thing, never use coarse adjustment unless you're on low, and let this clamp hold the slide in there so you never have to do it on your own. Okay, that's hopefully a good introduction so that you're ready to do the lab. The lab that we're going to do is one, the letter E, and you're going to notice it's upside down and backwards. There's some questions down there to figure out what the magnification power is. I'll give you a hint. This says 10 times, and that thing says 4 times. So I'll bet you can guess what the magnification is for the low power, the scanning part. Okay? And then when you go to the next strength of objective lens, you'll see, read it, and it'll tell you what it's written on there. It says A10. So this is 10 and that's 10. So there's a hint on what that one is. Notice what the next one is. I'm not going to read that one to you so you can do it on your own, and you're not going to use the oil one, but you are going to get to the high power one. Okay? So I got some questions for you that'll ask you what the magnification is and things like that. Read very carefully and hopefully you'll figure out, so I'm not just giving it away right here in this video. All right? So the thing that I want you to do in this lab is to look at the letter E and notice something cool. What happens to a letter E that's right side up when you look through a bunch of lenses, like a microscope or a telescope. And then I have some fibers over there that I might want you to look at, and I want you to notice how cool that is. I want you to play with it a little bit with the light and see if you don't get some more depth of field when you make it dimmer, when you kind of cut the light down, if you don't get a little bit more contrast, a little bit more of that. If you put the light bright on there, you might lose a lot of the color because you just white it out. But it depends which way you want it to be. So you get contrast or a more rich color depending on how you tweak the light. So play with it a little bit and see that you can see different things by doing that. Okay? Then we have a leaf from either an onion or a leaf from just a regular water plant. And you're going to take a tiny little slice out there, put it on the slide, right? Hopefully only 1 cell layer thick. And then you're going to put the slide on top and the dye that'll bring out those colors. Wait about 5 or 10 minutes and then look at that through the slide. Under low power first, and then you work your way up to the higher power. You're going to show your professor what it looks like that you did it under high power. So you're going to start with low, but then your professor will come around, look in there and see that, yeah, you got it. You got it under high power. The last thing is you're going to scrape some of the epithelial cells on the inner cheek. First you scrape it with the piece of wood, like a little toothpick first, and then just throw that one away. And then scrape that same spot over again so I'm not looking at your dinner or breakfast. I'm really just looking at your cells. Smear it right on the slide, put one drop of that dye on there, put the slide on top of it, maybe sideways a little bit, not straight on, but sideways so the bubbles go away.